So today, in terms of an overall agenda, as Janos mentioned, we'll talk a bit about molecular research through the lens of IP, the way we look at the world, and what we call our visual data revolution and the IP Geoscape tool that Janos mentioned. We'll go through some about the way we create the IP Geoscapes and the role that ChemCurator plays, the important role it plays in the development of our landscape technology. And then we'll talk about some machine learning and artificial intelligence issues looking forward in terms of Accencio and what we plan to do. So molecular research today, and I'll move fast, but please ask your questions and hopefully we can answer some of those throughout or at the end. As Janos mentioned, my background is in medicinal chemistry. I spent many years doing that before leaving and starting my legal career in Accencio. But one of the things that always perplexed me as a medicinal chemist, and one of the reasons for the creation of Accencio and everything that you're going to see today, is the fact that it was very difficult for me as a medicinal chemist and for chemists in general to really synthesize the vast amount of information that they need to synthesize while at the same time making molecules, generating them quickly, and understanding not only what they're doing, but being able to disseminate that information to others. I always say medicinal chemists had to be the smartest people in the room, because no one would ever come back to me or to the chemist and say, there's no freedom to operate. This is the molecule that you should make. Here, change this from a chlorine moiety to a fluorine moiety. Or no one would say, these compounds aren't active. This is the way that you could make them active. We had to understand the pharmacology, the biology, the legal aspects, everything else, and synthesize all that information. And one of the things that we discovered, Kevin Brogle and myself, before Accencio was created, is that there was no real way to synthesize all that information cohesively through one visualization to understand everything that was going on, including the issues associated with intellectual property. So we worked on things for quite some time, and our solution, really, at the end of the day, was to look at the world through an IP lens. And what I mean by that, very simply, is this. We wanted to use an understanding of intellectual property as the lens through which we viewed all other information, bring in all the necessary information for the chemist or for the scientist, the researcher, or whomever, that they would need to answer the questions they were looking to answer, but all of this through the lens of IP. And what does that mean specifically? And you'll see these 3 points, and I'll mention them throughout the presentation. But the first step is to create the lens, to understand in any given situation or for any given question what the IP to IP relationships are. And what I mean by that is the structural relationships. And if we're talking about large molecules, small molecules, composition of matter IP, you're looking literally at the relationships between structure A, structure B, and their similarity, as opposed to structure C, so on and so forth. But when we're talking about other types of IP, formulations, etc., you're looking at claim-to-claim relationships. But in any event, you're looking to understand firstly and create some sort of visualization that illustrates for you the IP to IP structural relationships. The second piece, very simply, is then to bring in all the other necessary information that you would need to answer the questions that you're seeking to answer. They could be related to the patents themselves, biological activity, predictive analytics, information about the companies, the assignees, the owners, anything from your perspective that you would need to answer questions that you're looking to answer. And finally, to use that information and the visualization itself and manipulate it using that other information to answer the questions you're looking to answer. Now, we at Accencio, we've created this platform of products that allows us or allows our customers to do that. And this is from everything, from early discovery all the way through commercialization and beyond, formulations, medical devices, small molecules, large molecules, whatever. It doesn't matter. It's always the same approach. Now, as this particular discussion is built around molecular research and seeing IP differently and using it and using visualizations and a look at IP differently to answer certain questions in a research environment, what I'm going to demonstrate for you today is our IP Geoscape tool. And I think you'll see very quickly the power therein. So let me just show you this very quickly. So what you're looking at is the first page of the IP Geoscape tool. And in this particular example, this IP Geoscape is built around the phosphodiesterase 9 target, so inhibition. And what you're looking at here, the number of patent families associated at this time when we created this particular landscape, there were 32 patent families from 11 different assignees and almost 4,000 exemplified molecules in this space. And we like to use this particular example for demonstration purposes because the number of molecules is low, the number of assignees is low, and you'll really get a feel for the power of the Geoscape and looking at things through an IP lens. But here are the 11 companies or assignees, at least at this time, that were operating in the space. There was one university, ASCA, Stellus, Bayer, Boehringer Ingelheim, so on and so forth. All of this is active. You can click on Bayer, for example. You can see their IP in the space. You can understand from a priority date perspective the amount of activity. This is on the left-hand side in the space. This is just a high-level view of who was doing what in the space, at least at the level of the patent. The real power of the landscape comes in is when you begin to look at the individual molecules associated with those particular assignees in the space. So let me explain to you what you're looking at. Every color here represents an individual company. So for example, you see here this red color, that's Bayer. This yellow color, for example, is Boehringer Ingelheim, excuse me. And each individual marker represents an exemplified molecule in a patent owned by that particular assignee. And you'll notice that some of the colors have more than one shape. All that simply indicates is that the particular assignee has more than one patent family in the space. So if, for example, we were to look at just Bayer in the space, you'll notice that there seems to be 1, 2, 3 different shapes. So that's 3 patent families for Bayer in the space. The lens that you're looking at here, these molecules are situated based on structural similarity. So if one molecule seems to be close to another molecule, that's because they're closely related structurally. And as you get further and further away, the molecules become more and more different from one another. So for example, here's a molecule from Ceylon, and here potentially is a molecule from Bayer. And you can see the similarity in the overall structure. So that's how you really interpret the map. So that is the overall lens. Now, when you're looking at the additional data that you can bring in right now, for example purposes, this is just a limited amount of data, but you can have information about the companies. For example, maybe you're only interested in academics. Maybe you're only interested in academics and private companies or others that may be there. There could be information about the companies. Maybe it's a licensing type of discussion and you're really interested in companies that really are highly capitalized in the space, and you can sort based on that type of information. At the company level, at the patent level, really an understanding of how the overall landscape developed over time from 2003, moving to 2005, on to 2008, so on and so forth. And then as a research scientist, for example, we have Lipinski's information in here, but for example purposes, if you have access to some sort of predictive analytics, and you really want to identify hotspots of activity, for example, one could really look to understand from a predictive perspective, what are the molecules predicted to be the most active in the space, and really begin to understand and look and see and say to oneself, well, if these are the most active compounds in the space, maybe I can, as a research scientist, begin to build around them, so on and so forth. And of course, from a medicinal chemistry perspective, the structure searching or substructure searching is always something that we as chemists like to do. And you can definitely do this here to be able to say to oneself, well, we've identified in our chemical inventory a particular moiety that seems to give rise to the greatest activity for us, and then begin to build your molecules around who else may be in the space with that type of structure, for example, and work your way around and get around certain patents, so on and so forth. So from an overall medicinal chemistry perspective, making new molecules, understanding what the competitors are doing, or even if you are, let's say, for example, we have other tabs here. Here's an analysis tab. Let's say, for example, you are Bayer and you want to understand your footprint in the space and who else may be operating in your space and the level of exclusivity, at least at the exemplified molecule level, for your products or your molecules in the space. And this is the same footprint. So this is Bayer's footprint, but it's just been reduced to these set of tiles. And anywhere you see a red tile, that's actually an area where Bayer has IP, but also some other companies have IP. And you can see what other companies are there. You can click on these links. You can go out to their patents. And what's more, you can go over to this medicinal chemistry tab here, and you could actually begin to understand in that particular tile what is or what may be the most common substructure, to begin to work your way around certain things or understand what structure or substructure there is giving rise to that amalgamation of compounds in that particular cluster of molecules. So, in the interest of time, that's just an example of the landscape and how you could potentially use it. And we create these, at least since we've been around, since 2017, for customers on a case-by-case basis. They would come to us and say, we have a particular target we'd like you to build a landscape around. We can make it, we can bring in whatever information they need, and they can use it for whatever their purposes may be. I'm happy to say that moving forward and launching next month, what we're going to be doing is launching our IP Geoscape library content. And what this will be is a series or a group of targets that we've picked out, and it's growing day by day. And we've picked them out for some obvious reasons: the level of activity in the space, how many patents or patent applications, are publishing every day. The velocity of activity in the area essentially gives rise to our list, and of course, we're working with our customers to add more targets that may be of interest to them. But you'll be able to purchase these, use them on your own, and come to certain decisions on your own without us just having to generate the landscape for you. So that's coming at the end of March. You'll see in maybe a few seconds there a little poll. If you can just click on which targets we have of our top 5 that you'd like to see, whichever target receives the most number of votes, I will actually go out and show you that target and walk through a couple of things with you just quickly to give you a feel for some newer targets and some things that may be of more interest to the group here. And I'll wait just another second or two to see if we get any request here. Any requests, Janos?
Yeah. This is the result. So our top performing one was epidermal growth factor receptor antagonists.
All right. So let's take a look at that. So this is EGFR, and if we go to this overview page, you can see now the vast number, the increase in the number of patent families, the number of assignees, the number of exemplified molecules. We're looking at over 17,000 molecules now. And you can also see why this was a target chosen over here on the left-hand side in terms of priority date. You can see the velocity and the amount of activity seems to definitely be increasing. And if we go to the landscape and we look, and we start to have a look around, for example, lots of activity going on here, but I can definitely show, this is the Amgen footprint, for example, and maybe we want to intersperse that with the Bayer footprint, for example, and maybe another company here, maybe BMS, to really begin to understand who's doing what and where. We can look at the molecules, for example, from Bayer, see what's going on, all the same things that you can do that I just illustrated for you in the PD9 landscape. So just by way of example, and just to show you the power that's here, looking at PD9 as opposed to looking at this particular landscape and trying to understand everything that's going on, filling in these black spaces with new molecules. If you're doing research and development, all of those things are possible using this type of landscape. So I just wanted to illustrate that for you quickly. So moving forward, one of the things we also wanted to talk about today is how ChemAxon and the ChemCurator product really helps us in generating our landscapes and the power of that product for our purposes. My partner Kevin Brogle is going to walk you through that process so you can understand it a bit better. But it's a very useful product here. So I'll pass it over to Kevin Brogle and he can talk to you about that.
Thanks, Brown. The process begins over on the left. You can see there with the selection of the target. And now this just involves more than just the protein, whether it be a receptor, an enzyme, or an ion channel. It includes the target concept as well. Are they activators, blockers, or are we going to incorporate both? Once that's determined, the relevant IP is identified. Now, those two steps are relatively straightforward, but at Accencio, in our IP Geoscape product, we look at IP relationships based on molecular structure. And because of this, we need to extract the structures from the patents. This step is not straightforward, or at least hadn't been until ChemCurator. ChemAxon's ChemCurator product, there highlighted in the middle, allows Accencio to efficiently, accurately, and economically extract the relevant structures, whether they occur in the form of names or in the form of images. And because of this, we can use our proprietary molecular quantization and mapping algorithms to produce the IP Geoscape visualizations that Kevin Brown has just shared with you. These visualizations that are then mineable in our Accencio View platform. So I'd like to, after that brief explanation of how we create our IP Geoscape visualizations and the role of ChemCurator, I'd like to hand it over to Árpád, who's going to tell you a little bit more about the ChemCurator product.
Thank you, Kevin. ChemCurator is created to speed up the extraction of high-quality chemical information from documents. So that means you may know a lot of different technologies available that allows you to bulk processing of large quantity of documents and extract information from them. However, we have the constant problem with these technologies that you are not able to trust the extracted information. If you want to get high quality extracted data, you need to involve some manual step into the process. ChemCurator gives you the combination of the two words. So speed up the process as much as possible with the automatic extraction techniques, however, allows the curator to fix unrecognized structures or misrecognized structures and do manual creation of the extracted information and enhancing the quality this way dramatically. ChemCurator is based on the listed technologies provided by ChemAxon, the English, Chinese, and Japanese name-to-structure technology, the structure search and representation that we have, and the Markush technology. In the described products, mainly the simple compound extraction use. However, during this presentation, I also show a brief introduction of the Markush extraction view. So the compound extraction view, it looks quite simple and straightforward. So in the middle of the screen, you can see the annotated document. That means you are able to load your documents into the ChemCurator application, and the application automatically recognizes all of the chemical entities inside the text and highlights you with gray. If you select any of these structures, it appears under the annotation view in the selected structures view. And from the selected structure view, the selected structures can be drag and drop and organized in different compound lists. Obviously, you are able to add additional information to the extracted compounds. So the original name, the preferred IUPAC name, and things like that automatically added to all of the extracted structure. However, you are able to add as many additional fields as many you want. For example, you are able to add the corresponding example number or any assay data that you want to record next to your extracted structures. In the Project Explorer, you can see your already loaded projects, so you can organize your work more efficiently this way. So this process helps a lot to extracting the chemical information. Or, of course, you are not only able to select and drag and drop structures that's recognized, but you are also able to select any unrecognized part of the text and add manually structures to it, or you are also able to change the extracted structures if you are not agree with the recognition. Here is how it works. So in the document, you are able to select the structures. These appears under the document. And from that view, you are able to drag and drop it to the compounds extraction view. The other important functionality of the application, the Markush extraction. Mainly in patent documents, the legal scope of the documents defined in the form of Markush claim. To extracting these Markush claims, a bit of more time consuming. However, it has a lot of advantage. You are able to define a searchable form, the complete legal scope of the document, and represent the complete chemical space covered by this legal scope. So the process is very similar. The ChemCurator only able to recognize you the different parts of the Markush claim and the chemical entities inside the Markush claim. You are who need to be ready to build up this recognized information in a form of Markush structure. However, we give you a quite intuitive UI where you can build up a tree-like hierarchy representation of the Markush structure, starting with the scaffold of the Markush structure and later to add the R-group definition and so on. During this process, you are also able to load the exemplified structures of the document under your Markush structure, and the tool doing an interactive checking of your example structures against the Markush structure. That means if any of your exemplified structures matching to the Markush structure highlighted by green, or if they are not matching, highlighted by red. The tool not only able to tell you these structures are matching or not, but also able to give you information about what are the matching parts of the Markush structures, or what are differentiating parts of the Markush structure, why it is not matching. One other important thing that I forgot to mention during the compound extraction, but also through here, all of the information that extracted and having the right-hand side linked to the original place in the document. So if you select any of the extracted structures, or if you select any of the extracted fragments from the Markush structure, the tool automatically scrolling to the corresponding part of the document. This is very useful if you need to double check your work, or you need to be sure that the information that you extracted is exactly what is in the document. Here is a brief overview about the capabilities of our Markush representation. So as you can see, the chemical representation part is quite rich. So most of the different Markush features that appears in a patent document can be easily represented by ChemAxon technology, and you are able to build up a Markush structure that is very, very similar to the original way how it's built up in the text of the document. As mentioned by Kevin, the ChemCurator application not only able to recognize the text-based information and the text-based chemical entities in the document, but using third-party chemical structure recognition tools, for example, Clyde and Ozra, we are also able to recognize the image-based chemical structures in the document. So depending on the quality of the document and depending on the drawing features, this can have a lot. You don't need to redraw this kind of structures from the based on the document, but you can use as a starting point, or as it recognized the information extracted from the images during the extraction process. As I mentioned, ChemCurator capable to handle different kinds of documents. These are including XML, PDF document, HTMLs. Obviously, text-based PDF documents works better than scanned PDF documents. We are also able to directly load documents from Google Patents based on the publication number, or if you have subscription to IFI claims based on the publication number, we are also able to directly download from IFN claims. So these are the capabilities of ChemCurator in a nutshell, and I would like to give back the word to Kevin. Thanks.
Thank you, Árpád. So just as a final slide in terms of the future role of machine learning and AI, at least in terms of what we're doing moving forward, at the end of the day, for us, it's about creating a repository of targeted IP data and really automating everything that you see here today. Currently, what we do is we create the landscapes and then we share them with the customers. They use them. But examples here include the automatic generation of new landscapes in target areas that are of interest to you, where you can put in the target, and the curation of the patents and the extraction of the molecule is something that we'll have the ability in the future to do in an automated fashion. And then also the automatic inclusion of your data or your molecules into the landscapes, where you can bring in one of our landscapes and you can upload an SDF file of your research molecules or certain data that you may know that no one else knows, but it's privy to you inside your company, to be able to sort through the landscape. So these are all things from an automation perspective that we plan to do, and you could look forward to very soon in the future from a machine learning and an artificial intelligence perspective. So with that being said, that is the end of the presentation. We thank you very much.